Approximately 155 million years ago, during the latter stages of the Jurassic period, the land masses that would eventually separate into modern continents remained largely connected, forming a single vast terrain of extraordinary biological density. The atmosphere carried higher concentrations of oxygen than the present day, and average global temperatures ran several degrees warmer than current baselines. These conditions did not merely permit large life, they actively sustained it, driving the metabolic demands of animals whose body masses would remain unmatched by any terrestrial predator in the history of life on Earth. The forests covering much of what is now western North America were dense, humid corridors of conifer species, araucarians, tree ferns, and cycads reaching heights of 30 to 40 meters and forming continuous canopies that filtered light into irregular, shifting columns. Ground-level visibility was limited. Moisture accumulated in low-lying terrain, producing persistent ground fog during cooler periods of the day. It was within this kind of environment, visually occluded, acoustically complex, and ecologically saturated, that the predators of the Jurassic operated. Before we go further, take a moment to consider where you're watching, from which country, and how distant it is from the Jurassic world we're about to enter, where enormous predators moved through landscapes far more dangerous than we once imagined. If these ancient worlds keep drawing you back, consider subscribing to continue the journey. It's always fascinating to see how far these stories travel. Lower the lights, let the present fade, and listen to the distant calls echoing through dense fern forests, where heavy footsteps move somewhere beyond the mist in a world that never truly felt safe. Among the most extensively documented of these predators is Allosaurus fragilis, a theropod dinosaur whose fossils have been recovered from the Morrison Formation in substantial numbers. Adult specimens reached lengths of between 8.5 and 12 meters, with body masses estimated between 1,500 and 2,000 kilograms in well-developed individuals. The skull was large relative to body size, but structurally lightened by a series of fenestrae openings in the bone that reduced cranial mass without significantly compromising integrity. Biomechanical analysis of jaw musculature and dental morphology indicates a hatchet-like strike pattern, in which the upper jaw was driven downward with considerable force against prey tissue, tearing rather than compressing. This feeding strategy suggests that Allosaurus was not primarily a bone processor, but a flesh-stripping predator whose attack cycle likely involved repeated strikes. Tooth marks recovered from sauropod bones in the Morrison Formation support this interpretation, consistent with a dragging motion, indicating flesh was pulled away from the skeleton rather than crushed at a single contact point. This behavior required prey that was either immobilized or physically constrained, which raises the central question of how Allosaurus consistently overcame animals that outweighed it by an order of magnitude. The prey available to large theropods of the late Jurassic was, in several cases, among the largest animals ever to walk on land. Diplodocus, Apatosaurus, and Camarasaurus all shared the Morrison ecosystem with Allosaurus, and all exceeded the predator's mass by factors ranging from 5 to more than 20. A fully grown Diplodocus could reach 25 meters in length and weigh between 10,000 and 16,000 kilograms. Even juveniles of these species, once past a certain size threshold, would have presented a dangerous and energetically costly target for a solitary predator. A single strike from a sauropod tail or forefoot could cause skeletal injury sufficient to compromise a predator's long-term survival. Evidence from the fossil record indicates that Allosaurus sustained such injuries with regularity. Healed fractures and pathological bone remodeling have been identified in multiple specimens, concentrated in the ribs, vertebrae, and forelimbs, consistent with blunt force trauma from large prey animals. The fact that healing is evident confirms these animals survived initial trauma, but also indicates that injury was a recurring occupational condition rather than a rare exception. Hunting in this ecological context carried measurable physical cost that accumulated across the lifetime of individual predators. At the Cleveland Lloyd Dinosaur Quarry in Utah, the remains of at least 46 individual Allosaurus specimens have been recovered from a single stratigraphic horizon, one of the densest concentrations of theropod material known from the Jurassic. One hypothesis proposes predator trapping at a seasonal water source in which large herbivores became mired in sediment and attracted multiple predators over an extended period, some of which also became trapped. If this interpretation holds, it implies that multiple Allosaurus individuals were simultaneously drawn to the same prey resource, a pattern consistent with tolerance of conspecifics near food, if not active cooperation. Whether this tolerance extended to coordinated predation remains unresolved. Some researchers propose that loose theropod aggregations may have pressured prey directionally, reducing escape options without requiring the behavioral synchrony seen in modern social hunters. Others maintain the evidence supports nothing beyond opportunistic co-feeding. The distinction is significant. Even rudimentary coordination would meaningfully alter the threat profile that large theropods presented to the ecosystem's largest herbivores. The temporal distribution of predatory activity cannot be directly recovered from the fossil record, but scleral ring analysis using bony eye structures that provide indirect evidence of pupil geometry has been applied to extinct theropods to model activity patterns. Analysis suggests that large theropods, including Allosaurus, showed proportions consistent with cathameral behavior, meaning activity distributed across both day and night rather than confined to a single window. If accurate, predation pressure in the Jurassic extended across multiple periods of the daily cycle rather than concentrating at predictable intervals. The dusk period, in particular, represents a transitional zone of ecological consequence. Light levels drop rapidly, reducing visual acuity in prey animals with cone-dominated retinal structures optimized for full daylight. A predator functioning effectively in lower light conditions would gain a measurable advantage during this interval. In forested environments with dense canopy cover, ambient light degrades faster than in open terrain, meaning the structural density of late Jurassic forests interacted directly with predator sensory biology to produce specific windows of elevated risk. Stegosaurus, which shared the Morrison Formation with Allosaurus, possessed a tail-mounted arrangement of paired spikes whose geometry and preserved damage patterns indicate active defensive use. Puncture marks consistent with Stegosaurus tail spikes have been identified on Allosaurus specimens, confirming direct physical contact and confirming that predation attempts on Stegosaurids carried quantifiable injury risk. The ecological relationship between these two species was one of reciprocal physical pressure, a dynamic in which both parties sustained damage and in which the outcome of individual encounters was far from predetermined. Diplodocus tail morphology presents a parallel case. The elongated, whip-like caudal vertebrae of this genus have been analyzed as a potential defensive structure capable of generating supersonic velocity at the distal tip, producing both acoustic deterrence and direct physical threat. The Jurassic predator, therefore, did not occupy a position of unchallenged dominance. It operated within a system of competing pressures: prey resistance, conspecific competition, environmental constraint, and the constant metabolic demand of sustaining a large body in a resource-limited world. What the fossil record preserves in bone, in track, in tooth mark, is not the drama of individual moments, but the cumulative signature of a system under continuous pressure. The fog-covered forests, the dense vegetation, the volcanic horizons visible at distance, these were not passive backdrops. They were active variables shaping the outcome of every predation attempt. Visibility determined detection range. Terrain determined pursuit viability. Humidity and temperature influenced metabolic urgency and therefore the frequency of necessary feeding events. The predators of the Jurassic moved through an environment that offered neither consistent advantage nor reliable safety to any species within it. Size was not a guarantee of survival. It was the opening condition of a far more complex ecological equation, one being continuously resolved across millions of years of reciprocal pressure, adaptation, and constraint. During the late Jurassic period, approximately 150 to 155 million years ago, the terrestrial ecosystems of what is now North America and parts of Europe supported a concentration of large-bodied herbivores without parallel in the subsequent fossil record. These were the sauropods, long-necked, quadrupedal dinosaurs whose body plans represented the most extreme expression of large body size ever achieved by a land-dwelling vertebrate. They did not simply inhabit the Jurassic landscape. At their scale, they actively restructured it. The physical dimensions of these animals remain difficult to contextualize against modern reference points. Brachiosaurus altithorax, one of the more massive species recovered from the Morrison Formation, is estimated to have reached lengths of 26 meters and body masses of between 30,000 and 60,000 kilograms in large adults. Though mass estimates for sauropods carry considerable uncertainty due to the body masses of SDP. Difficulty of reconstructing soft tissue volume from skeletal remains alone. Diplodocus carnegie reached comparable lengths, sometimes exceeding 25 meters, though with a more slender body plan and correspondingly lower mass. Apatosaurus ajax was shorter but more robustly constructed, with vertebral and limb proportions suggesting a particularly dense and heavily muscled body. Each of these species occupied the same broad ecosystem simultaneously, which raises immediate questions about how a single environment sustained animals of this magnitude in sufficient numbers to persist across geological time. The answer lies partly in the productivity of the late Jurassic environment itself. The Morrison Formation, which preserves the remains of these animals across a geographic area exceeding 1 million square kilometers, accumulated during a period of relatively high atmospheric carbon dioxide concentration. Current estimates place Jurassic CO2 levels at between 3 and 8 times pre-industrial values, a condition that significantly elevated plant growth rates and biomass production across vegetated surfaces. The forests and floodplains of the period generated organic material at rates sufficient to support large populations of high-consumption herbivores, but only if those herbivores could process and extract energy from plant material efficiently. Sauropod feeding biology was adapted to exactly this requirement. Rather than thoroughly chewing plant material, a metabolically slow process, sauropods appear to have cropped and swallowed vegetation with minimal oral processing, relying on fermentation within a large gut volume to extract nutrients over an extended digestive period. Tooth wear patterns in species such as Diplodocus indicate a raking or stripping motion, pulling leaves and soft vegetation from branches rather than grinding tough fibrous material. Camarasaurus, by contrast, shows more robust dental wear consistent with processing harder or coarser plant matter. This divergence in feeding mechanics between coexisting sauropod species is functionally significant. It indicates resource partitioning, in which different species exploited different components of the available vegetation, reducing direct competition and allowing multiple large-bodied herbivores to persist within the same geographic range. The energetic requirements of animals at this scale were continuous and substantial. A sauropod of 30,000 kilograms body mass would have required daily caloric intake far exceeding that of any modern terrestrial herbivore. Metabolic modeling based on bone growth rate analysis using growth rings preserved in sauropod cortical bone suggests that these animals grew rapidly during juvenile stages, reaching sexual maturity within 1 to 2 decades, and continuing to add mass for years beyond that. Rapid growth rates are generally associated with elevated metabolic activity, implying that at least some sauropod species maintained higher body temperatures and faster energy processing than cold-blooded ectotherms of comparable size. This interpretation remains debated, but the bone histology data consistently indicates growth trajectories incompatible with the slow, seasonally interrupted growth patterns typical of modern reptiles. To sustain this metabolic demand, sauropods fed across extended daily periods. The long neck, the most anatomically distinctive feature of the sauropod body plan, functioned as a feeding reach multiplier, allowing an animal to access vegetation across a large arc without repositioning its enormous body mass. Brachiosaurus, with a relatively upright neck posture, could browse at heights of up to 9 meters above ground level, accessing canopy vegetation unavailable to shorter-necked competitors. Diplodocus and Apatosaurus, with necks oriented more horizontally, swept vegetation at lower heights across a wide lateral range. In both cases, the neck extended the effective foraging radius of the animal without requiring the energetic cost of locomotion, a significant efficiency advantage for an animal whose movement itself demanded considerable caloric expenditure. Locomotion in animals of sauropod mass was governed by physical constraints that shaped both their behavior and their ecological footprint. Each footfall of a large sauropod transferred forces of tens of thousands of newtons into the substrate. On saturated floodplain soils, the depositional environments where most Morrison Formation fossils are preserved, this pressure was sufficient to displace substrate to depths of several centimeters per step, leaving tracks that were subsequently preserved in fine sediment. Sauropod trackways recovered from Jurassic formations in North America, Europe, and Africa confirm that these animals moved in consistent directional patterns, often in groups, across terrain that bore the physical impression of their passage long after they had moved on. The cumulative effect of large sauropod herds moving through a forested environment would have been considerable. Mature trees, broken or uprooted by the passage of animals weighing tens of thousands of kilograms, would have created gaps in the canopy, allowing light to reach the forest floor and stimulating the growth of lower-lying vegetation. This disturbance-driven regeneration cycle in which large herbivores destroyed mature plant structures and inadvertently promoted new growth is a pattern observed in modern elephant-dominated ecosystems, where mega-herbivore activity maintains a mosaic of vegetation types that would otherwise converge toward closed canopy forest. In the Jurassic, sauropods may have performed an ecologically analogous function at a scale an order of magnitude larger. Migration, or some form of seasonal directional movement, is hypothesized for several sauropod species based on isotopic analysis of tooth enamel. Oxygen and strontium isotope ratios preserved in tooth material reflect the geochemical signature of water sources consumed during the period of tooth formation. In some specimens, isotopic values shift across the growth sequence of a single tooth in patterns consistent with movement between geographically distinct water sources implying seasonal displacement of hundreds of kilometers. If confirmed by broader sampling, this would indicate that sauropod populations were not static occupants of fixed home ranges, but mobile consumers tracking seasonal resource availability across large portions of the Morrison landscape. Such movement would have had cascading ecological consequences. Herds of animals, each weighing tens of thousands of kilograms, moving across floodplains and through forested corridors on seasonal schedules, would have transported nutrients across geographic boundaries, disturbed soil and vegetation at regular intervals along consistent routes, and concentrated predator activity at predictable locations where herbivore density was highest. The sauropod migrations of the late Jurassic, to the extent that they occurred, were not merely biological events, they were geological and ecological processes, reshaping terrain and redistributing resources across a landscape that responded visibly to their passage. What the sauropods represented, ultimately, was not merely a biological extreme, but an ecological threshold, a point at which individual organisms became significant variables in the function of entire landscapes. Their feeding altered forest structure, their movement redistributed soil and nutrients, their presence sustained predator populations whose own size was calibrated to the scale of available prey. The ecosystem of the late Jurassic did not contain these animals incidentally. It was, in measurable ways, organized around them, shaped by their requirements, disturbed by their movement, and dependent on their continued presence for the maintenance of conditions that sustained the broader community of species living alongside them. While terrestrial ecosystems of the late Jurassic supported life at scales difficult to contextualize against modern reference points, the oceans of the same period presented a parallel but distinctly different expression of biological extremity. The shallow epicontinental seas that spread across much of what is now Europe and the deeper marine corridors connecting ancient ocean basins were inhabited by predators whose body plans, sensory systems, and feeding behaviors were calibrated to an aquatic environment with no surviving equivalent. These were not marginal or transitional creatures. They occupied apex positions in marine food webs that had been developing and intensifying for tens of millions of years. The late Jurassic oceans were warmer than present-day seas by an estimated 5 to 10 degrees Celsius at surface level, with reduced polar ice and correspondingly higher global sea levels. This thermal regime supported high marine productivity-abundant plankton, dense populations of invertebrates, and large schools of fish and cephalopods that form the nutritional foundation of the ecosystem. Into this productive environment, multiple lineages of large marine reptiles had independently evolved, each exploiting different components of the available prey base through distinct anatomical and behavioral strategies. Among the most formidable of these marine predators were the pliosaurs, short-necked, large-skulled plesiosaurs whose body plan was oriented entirely toward high-speed pursuit and powerful bite delivery. Leopleurodon ferox, recovered primarily from late Jurassic deposits in England and France, represents one of the better documented large pliosaur species of the period, though size estimates have varied considerably and earlier figures of 25 meters have been revised downward by subsequent analysis. Current conservative estimates place large Leopleurodon specimens at 6 to 7 meters in length, with some fragmentary material suggesting individuals approaching 10 meters. The skull alone in large specimens accounted for a substantial proportion of total body length, housing conical teeth adapted for seizing and retaining mobile prey rather than cutting or processing it. Pliosaur locomotion was driven by 4 large paddle-shaped limbs generating thrust through a flying motion analogous to that of modern sea turtles, but scaled to a body mass that biomechanical modeling suggests could exceed 1,000 kilograms in large individuals. This propulsion system was capable of generating rapid acceleration over short distances, a functional profile consistent with ambush or short pursuit predation rather than sustained open water chase. The narial anatomy of pliosaurs, with internal nostril placement allowing water to be drawn through the olfactory system during forward movement, suggests that chemoreception played a significant role in prey detection at range, with visual targeting becoming primary only during the final approach. Sharing these waters was a second and anatomically contrasting lineage, the ichthyosaurs. By the late Jurassic, ichthyosaurs had occupied marine environments for more than 100 million years and had converged on a body plan strikingly similar to that of modern dolphins and tuna, streamlined, fast, and built for sustained locomotion in open water. Ophthalmosaurus eisenicus, one of the most abundant ichthyosaur species of the late Jurassic, possessed eyes with a diameter of up to 23 centimeters, among the largest relative to body size of any known vertebrate. The structural reinforcement of the eye by a ring of scleral ossicles capable of maintaining shape under pressure indicates that Ophthalmosaurus was adapted for vision in low light or deep water conditions. This anatomical evidence positions Ophthalmosaurus as a deep-diving predator capable of descending to depths where ambient light was severely reduced and pursuing prey, primarily cephalopods and fish, in conditions that would impair the visual performance of shallower adapted predators. Oxygen storage capacity, inferred from body volume and diving physiology models, suggests dive durations potentially exceeding 20 minutes. At depth, in near darkness, Ophthalmosaurus represented a predator operating at the boundary of what the marine environment physically permitted, exploiting a resource zone largely inaccessible to surface-oriented competitors. The cephalopod fauna of the late Jurassic oceans was itself extraordinary in its diversity and abundance. Ammonites shelled cephalopods with coiled, chambered shells existed in hundreds of species during this period, ranging in shell diameter from a few centimeters to, in later geological periods, exceptional specimens exceeding 1.5 meters. In the late Jurassic, ammonite populations formed dense aggregations in shallow marine environments, drifting in the water column and feeding on plankton and small invertebrates. Their abundance made them a primary prey resource for multiple marine predator lineages simultaneously, and their shells, preserving exceptionally well in fine marine sediment, now constitute some of the most common and biostratigraphically useful fossils of the Mesozoic. Belemnites, a related cephalopod group with an internal shell and a squid-like body plan, were similarly abundant and likely formed schooling aggregations that attracted large predators in the manner of modern fish schools. Evidence for direct predation on belemnites comes from gut content preservation in ichthyosaur specimens, where the mineralized rostral guards of belemnites, the dense, bullet-shaped internal structures, have been recovered in concentrated masses within the rib cage of the predator. In some specimens, hundreds of individual belemnite guards are preserved together, indicating that single feeding events involved the consumption of large numbers of prey items rather than individual pursuit of isolated targets. Above the surface of these seas, conditions were rarely stable. The late Jurassic atmosphere, driven by elevated ocean temperatures and active volcanic systems along mid-ocean ridges and continental margins, generated weather patterns of considerable intensity. Tropical storm systems moving across shallow epicontinental seas would have produced wave heights and surface turbulence sufficient to affect the behavior of even large marine animals, forcing deeper water movement and disrupting surface feeding activity. The interaction between atmospheric instability and marine biology was not incidental. Storm-driven upwelling along coastlines brought nutrient-rich deep water to the surface, temporarily increasing prey density in shallow zones and drawing large predators inshore. These coastal margins were ecologically dynamic and physically unstable. Where rivers discharged into shallow marine basins, freshwater lenses formed above denser saltwater, creating stratified zones of variable salinity that some species tolerated and others avoided. Fine sediment carried by river systems accumulated in these nearshore environments, preserving the remains of marine organisms in exceptional detail when conditions of rapid burial and low oxygen inhibited decomposition. The extraordinary fossil preservation observed in several European late Jurassic deposits, including the Solnhofen limestone of Bavaria, reflects precisely these conditions. Calm, hypersaline lagoonal environments adjacent to open marine waters, where organisms sinking to the substrate were buried before scavengers or decay could disarticulate them. The marine ecosystems of the late Jurassic were not characterized by any single dominant predator, but by a layered structure of competing lineages, each occupying distinct but partially overlapping ecological roles. Pliosaurs pursued large, mobile prey in nearshore and open water environments. Ichthyosaurs exploited deep water and low-light niches inaccessible to surface-adapted competitors. Large sharks, including hybodont and neoscalation forms, processed prey through repeated biting rather than whole capture, occupying a scavenging and opportunistic predation role alongside active hunters. Marine crocodilians, present in the late Jurassic and adapted for saltwater tolerance, occupied coastal and estuarine zones where terrestrial and marine food webs intersected. The result was an ocean in which predation pressure operated simultaneously across depth zones, light conditions, and prey size, classes a vertically and horizontally integrated system of consumption whose complexity exceeded that of most modern marine environments. The animals moving through these waters did so within a competitive structure that demanded continuous energetic output, precise sensory function, and behavioral flexibility. Survival in the Jurassic Ocean was not secured by size or speed alone. It was the product of an ongoing intersection between biological capability and an environment that offered resources abundantly but distributed them unpredictably across a vast, dark, and physically unforgiving medium. The terrestrial forests of the late Jurassic were not passive environments. They were dense, biologically saturated systems in which every vertical layer from the root networks beneath the soil to the upper canopy 30 to 40 meters above ground level was occupied by organisms competing for resources, evading predation, or actively hunting. The structural complexity of these forests created conditions in which danger was not concentrated at predictable points, but distributed continuously across space and time. For any animal moving through this environment, the variables were constant and the margins were narrow. The dominant vegetation of the period consisted primarily of conifers, cycads, tree ferns, and horsetails plant lineages that had developed across hundreds of millions of years prior to the Jurassic and had reached high levels of structural and chemical sophistication. Many conifer species produced resins and secondary compounds that deterred insect feeding. Yet insect diversity in the late Jurassic remained extraordinary, with thousands of species occupying decomposer, herbivore, predator, and parasitic roles simultaneously. The forest floor accumulated deep layers of organic debris, fallen wood, leaf litter, shed scales and fronds that retained moisture and maintained the humid microclimatic conditions in which a large proportion of small-bodied fauna lived and reproduced. Insects of the late Jurassic reached body sizes considerably larger than most modern equivalents in several lineages. Elevated atmospheric oxygen levels, which enhance the efficiency of the tracheal respiratory systems insects rely upon, permitted larger body volumes in groups including dragonfly relatives and orthopteran ancestors. Some reconstructed specimens from broadly contemporaneous deposits indicate wingspans or body lengths several times those of their nearest living relatives. In this forest environment, insects were not merely background fauna, they were ecologically significant in their aggregate biomass as vectors of disease and parasitism, and as prey resources that sustained entire guilds of small vertebrates. Parasitic invertebrates, including early representatives of groups ancestral to modern mites and biting flies, were present in Jurassic ecosystems and are preserved in amber deposits from broadly contemporaneous and slightly later periods. The presence of blood-feeding ectoparasites implies that large-bodied dinosaurs sustained persistent parasite loads, a condition with measurable consequences for immune function, energy allocation, and long-term health. An animal expending metabolic resources on immune response to chronic parasitism is simultaneously constrained in its capacity for growth, reproduction, and sustained physical activity. In a competitive ecosystem where energetic margins were already narrow, parasite pressure represented a continuous and invisible cost. The forest understory, the zone between the ground and the lower canopy, was where the greatest density of ecological interaction occurred. Small and medium-bodied dinosaurs navigated this layer in search of food, shelter, and reproductive opportunity, while simultaneously managing the risk of encounter with larger predators moving through the same space. Ornithopod dinosaurs of modest body size, early ceratopsians, and small theropods all occupied this zone, relying on sensory acuity, rapid locomotion, and behavioral familiarity with local terrain to reduce predation exposure. The vegetation itself created both concealment and constraint. Dense cycad growth and low-branching conifers provided cover that disrupted sightlines and reduced the effective detection range of both predator and prey. An ambush predator positioned within dense vegetation could reduce its detection distance to a few meters, well within the reaction time window of many prey species. Conversely, prey animals moving through the same vegetation were navigating a continuous series of potential concealment points, each of which represented a location from which an attack could originate. The spatial structure of the forest was not neutral. It actively shaped the probability distribution of predation events. Wetland margins, which were extensive across the Morrison Formation landscape, and equivalent Jurassic environments elsewhere, introduced additional hazard variables. Standing water of shallow depth supported populations of large amphibians, early crocodilians, and aquatic invertebrates, while the soft substrate surrounding wetland margins preserved tracks but also created footing instability for large animals. A large herbivore moving through saturated ground near a water source was simultaneously exposed to attack from terrestrial predators and constrained in its ability to accelerate or change direction rapidly. Wetland margins concentrated animal activity because water was a non-negotiable resource requirement and simultaneously degraded the locomotor performance that constituted a primary escape mechanism. Fog accumulated preferentially in low-lying terrain, including river valleys and wetland basins, during the cooler hours preceding dawn. In these conditions, visibility could fall to tens of meters within enclosed forest, reducing the detection advantage of visually oriented predators, but also eliminating the early warning distance that prey animals depended upon to initiate escape behavior. The convergence of reduced visibility, constrained locomotion, and concentrated animal activity at wetland margins produced conditions of elevated predation risk during specific daily intervals, a pattern that, if recognized behaviorally by either predator or prey, would have generated predictable temporal and spatial clustering of dangerous encounters. Beneath the movements of large dinosaurs, the forest floor sustained a community of small-bodied vertebrates whose survival strategies were oriented almost entirely around avoiding detection. Early mammals of the late Jurassic, multituberculates, triconodonts, and related lineages were predominantly small, with body masses in the range of a few grams to several hundred grams. Evidence from tooth morphology, limb proportions, and the geometry of preserved burrow structures indicates that many of these animals were nocturnal and fossorial, spending daylight hours in underground chambers and emerging to forage during periods of reduced predator activity. For these animals, the forest was experienced at ground level and below a landscape of root systems, leaf litter depth, soil moisture gradients, and the sound and vibration signatures of large animals moving overhead. A sauropod footfall transmitted ground vibration detectable at distances of tens of meters, providing indirect positional information about the movement of animals too large to be encountered safely. The forest floor fauna had, over millions of years, developed sensory and behavioral frameworks calibrated to this specific informational environment, one in which direct visual contact with a threat was often preceded by acoustic or seismic signals that permitted preemptive concealment. Humidity in the late Jurassic forest was not merely an atmospheric condition, it was a biological medium. High moisture levels accelerated the growth of fungi and microbial communities that broke down organic material rapidly, recycled nutrients through the soil system, and sustained the plant productivity on which the entire food web depended. It also created conditions favorable to the transmission of pathogens. Wounds sustained during predation attempts or intraspecific conflict in an environment of high microbial activity and warm temperatures would have been subject to rapid infection. Bone pathologies identified in Jurassic dinosaur specimens, abnormal growths, erosive lesions, and evidence of chronic infection confirm that disease and injury-related deterioration were significant mortality factors operating alongside direct predation. The late Jurassic forest, considered as an integrated system, was dangerous not through the presence of any single overwhelming threat, but through the accumulation and interaction of multiple simultaneous pressures. Predators of varying size operated across all temporal windows. Parasites and pathogens imposed continuous physiological cost. The physical structure of the environment constrained movement and reduced sensory range. Water sources concentrated animals under conditions of impaired escape capacity. These pressures did not operate in isolation, they intersected, reinforced one another, and produced a cumulative risk environment that no species navigated without cost. Survival here was not a stable condition. It was an outcome that required continuous, precise biological performance across every dimension of an organism's existence. The transition from daylight to darkness in the late Jurassic was not a neutral event. It was an ecological shift, a reconfiguration of the sensory landscape that redistributed advantage and vulnerability across the entire community of organisms sharing a given environment. As light levels fell below the threshold required for cone-based photopic vision, animals whose sensory systems were calibrated primarily for daylight operation experienced a measurable degradation in their ability to detect, track, and respond to movement. For other animals, whose anatomical investments had been directed toward low-light performance, this same transition represented an expansion of functional capacity. The Jurassic night was not simply the absence of day. It was a distinct ecological state, inhabited by a specific suite of organisms whose survival depended on their ability to operate within it. The physical conditions of the late Jurassic night were shaped by several interacting variables. Cloud cover, which was frequent given the elevated atmospheric moisture and active weather systems of the period, could reduce ambient light levels to near zero on overcast nights, eliminating even the contribution of stellar and lunar illumination. Conversely, on clear nights at the latitudes occupied by the Morrison Formation ecosystem, lunar illumination would have provided sufficient ambient light for scotopic vision, the rod-based low-light visual system present in most vertebrates, to function across detection ranges of several tens of meters in. Open terrain. Within dense forest, these ranges contracted sharply as canopy cover intercepted available light and produced a ground-level environment of near-total darkness even on clear nights. Theropod dinosaurs present the most complex and debated case with respect to nocturnal activity. Scleral ring analysis, applied to theropod specimens where preservation permits, has suggested cathemeral activity patterns in some large-bodied species, activity distributed across both day and night without strict confinement to either. This interpretation is consistent with the metabolic demands of large predators, whose energetic requirements necessitate feeding opportunities across extended daily periods rather than within the narrow window of a single light regime. A predator capable of functioning across the full daily cycle would be unconstrained by the temporal behavior patterns of prey species that retreated to specific environments at night, providing access to a broader range of foraging opportunities. Small theropods present a partially different picture. Megapnosaurus and related coelophysoid forms, along with early members of lineages that would later diversify significantly, show skeletal proportions and inferred sensory investments consistent with active pursuit predation in variable light conditions. The relative orbit size in some small theropod skulls, a proxy for eye volume and therefore light-gathering capacity, is proportionally larger than in some larger relatives, suggesting elevated investment in visual performance under low-light conditions. These animals likely targeted the small vertebrates and invertebrates that became more accessible at night, when the concealment behaviors of diurnal prey species were less effectively maintained. The mammals of the late Jurassic navigated the nocturnal environment with sensory systems that were, by this period, already substantially refined for low-light operation. The mammalian lineage had passed through an extended period of evolutionary history under sustained predation pressure from archosaurs, and the sensory and behavioral architecture of late Jurassic mammals reflected this history directly. Relative brain size in early mammals was elevated compared to reptilian contemporaries of equivalent body mass, with disproportionate investment in the olfactory and auditory processing regions, sensory modalities that function independently of ambient light. Hearing in early mammals was enhanced by the progressive detachment of the quadrate and articular bones from the jaw joint elements that in mammalian ancestors had served a structural role in the skull, but in mammals were being incorporated into the middle ear as additional ossicles. This anatomical transition, documented in the fossil record across multiple mammalian lineages of the Mesozoic, significantly improved the sensitivity and frequency range of auditory detection. A small mammal foraging on the forest floor in near-total darkness could construct a detailed acoustic map of its immediate environment, tracking the movement of invertebrate prey, detecting the approach of predators through substrate vibration and airborne sound, and maintaining spatial orientation without any dependence on visual input. Olfaction provided a complementary and equally critical sensory channel. Chemical signals distributed through the environment, scent marks deposited by other animals, volatile compounds released by disturbed vegetation, the odor signatures of predators moving upwind provided continuous informational input that visual darkness did not interrupt. Early mammals invested heavily in olfactory epithelial surface area. as evidenced by the complexity of preserved nasal terminal structures in well-preserved specimens. The result was a sensory system capable of detecting and discriminating between chemical signals at concentrations far below the thresholds detectable by most contemporary reptilian predators, providing an informational advantage that partially compensated for small body size and limited locomotor speed. Burrow systems represented the primary structural defense available to small-bodied nocturnal mammals. Underground chambers, accessed through narrow entrance tunnels that excluded larger predators, provided thermal stability, humidity regulation, and protection from surface-level predation during periods of rest. The geometry of burrow entrances, typically sized to permit passage of the resident animal while excluding larger-bodied threats, created a physical security threshold that required no active behavioral maintenance during occupation. Evidence for fossorial behavior in Mesozoic mammals comes from limb proportions consistent with digging, from the preservation of individuals in curled postures suggestive of underground rest, and from trace fossil evidence of small-diameter burrow systems in Mesozoic terrestrial deposits. The soundscape of the Jurassic night carried information that extended well beyond the immediate sensory range of any individual animal. Large theropods moving through forest vegetation at night displaced substrate and vegetation in patterns detectable as low-frequency sound and ground vibration at distances considerably exceeding direct visual range. The calls of animals, whether territorial, communicative, or involuntary stress vocalizations, propagated through the forest in ways that were shaped by vegetation density, terrain topography, and atmospheric conditions. Sound traveling along valley floors or across open water surfaces carried farther and with less attenuation than sound moving through dense forest, creating acoustic corridors that connected distant portions of the landscape. Whether Jurassic dinosaurs produced vocalizations and of what character cannot be directly determined from skeletal remains alone. Soft tissue vocal structures do not preserve in the fossil record under typical conditions. However, phylogenetic bracketing, inferring the likely characteristics of extinct animals from the known biology of their living relatives, indicates that archosaurs broadly possess the capacity for vocalization, as evidenced by the calls of modern crocodilians and birds, the two living archosaur lineages. The specific acoustic properties of any Jurassic dinosaur vocalization remain irrecoverable, but the structural capacity for sound production was almost certainly present across multiple lineages, implying that the nocturnal environment was not acoustically silent, but populated with signals whose interpretation was itself a survival-relevant cognitive task. The Jurassic night, considered as an ecological system, was not a simplified version of the day with fewer participants. It was a fully operational biological environment in which different species, different sensory modalities, and different behavioral strategies came to the foreground. The animals that inhabited it successfully had done so through specific and costly anatomical investments in eye structure, in auditory ossicle geometry, in olfactory surface area, in the musculoskeletal architecture of digging. These investments were not incidental. They were the accumulated product of selection pressure operating continuously across millions of years, in an environment where the consequences of sensory or behavioral failure were immediate and irreversible. While the terrestrial and marine environments of the late Jurassic were dominated by animals whose scale and predatory capacity have few modern equivalents, the aerial dimension of this ecosystem was occupied by a lineage whose body plan represented one of the most consequential evolutionary innovations of the Mesozoic. Pterosaurs, flying reptiles whose origins extend back to the Triassic period, had by the late Jurassic diversified into a range of ecological roles that included coastal fishing, inland scavenging, pursuit predation, and filter feeding. They were not primitive or transitional forms. By 150 million years ago, pterosaurs had been refining their flight apparatus for more than 50 million years, and the animals present in late Jurassic skies were sophisticated flyers whose aerodynamic performance current biomechanical modeling continues to reassess upward. The pterosaur wing was constructed around a dramatically elongated fourth digit, from which a membrane of skin and internal fibers, the patagium, extended to the body and, in most species, to the hind limb. This membrane was not a simple passive surface. Fossil preservation in exceptionally fine-grained deposits, including the Solnhofen limestone of Bavaria, reveals internal structural fibers called actinofibrils running through the wing membrane at consistent angles, providing stiffness regulation that the animal could modulate through muscular control of the attached limb elements. The result was a wing capable of adjusting its camber and surface tension in response to changing aerodynamic conditions, a level of active flight control that placed pterosaurs well beyond the mechanical simplicity of a passive gliding membrane. Among the late Jurassic pterosaurs, Rhamphorhynchus munsteri is among the most completely known, with numerous specimens recovered from the Solnhofen deposits preserving not only skeletal material but soft tissue impressions of the wing membrane, cranial crests, and, in some cases, the outline of a throat pouch. Rhamphorhynchus possessed a long tail terminating in a diamond-shaped vein, which functioned as a stabilizing surface during flight, providing pitch and yaw control that compensated for the aerodynamic challenges presented by the animal's relatively large head. Wingspan in adult specimens reached approximately 1.8 meters, placing Rhamphorhynchus within a size range compatible with sustained soaring over coastal water surfaces, where updrafts generated by the interaction of wind with cliff faces and wave surfaces provided lift with minimal energetic expenditure. The dentition of Rhamphorhynchus, long, forward-angling teeth interlocking when the jaw was closed, is consistent with a fish-seizing feeding strategy, in which the jaws were plunged into the water surface during low flight to intercept prey near the surface. This feeding mode, analogous to that of modern skimmers, requires precise coordination between flight speed, jaw angle, and water surface contact, and implies a level of sensory integration and motor control sophisticated enough to execute a high-speed interaction with a moving and unpredictable medium. Fish scales and bones have been recovered from the stomach regions of preserved Rhamphorhynchus specimens, confirming the dietary inference derived from dental morphology. Larger pterosaurs shared the late Jurassic aerial environment. Pterodactylus antiquus, recovered from the same Solnhofen deposits, represented a distinct body plan, short-tailed, with a proportionally larger skull and a more flexible neck that is considered more aerodynamically advanced than the long-tailed rhamphorhynchoid form. Wingspan estimates for Pterodactylus range from under 1 meter in juveniles to approximately 1.5 meters in large adults, though the species has historically been applied to a range of specimens now recognized as representing multiple distinct taxa. The postcranial proportions of pterodactyloid pterosaurs generally indicate a more terrestrially capable animal than their long-tailed relatives. Limb geometry and joint orientation suggest that pterodactyloids could fold their prophyses, wings compactly, and move quadrupedally on solid surfaces with reasonable efficiency, expanding their foraging range beyond the water margins that long-tailed forms appear to have favored. Nesting behavior in pterosaurs is documented by egg and embryo material recovered from several Mesozoic deposits, though late Jurassic nesting evidence specifically remains limited. The eggs of pterosaurs were leathery and parchment-like rather than hard-shelled, implying burial in moist substrate rather than incubation in open nests, a strategy that reduced exposure of eggs to aerial predators but required selection of substrate with appropriate thermal and moisture properties. Nesting sites on cliff faces, coastal dunes, or vegetated inland areas would have represented significant concentrations of vulnerability, where adults returning to tend eggs or hatchlings were predictably located, and where the eggs themselves were accessible to ground-level scavengers capable of locating buried organic material through olfaction. The weather systems of the late Jurassic presented a persistent physical challenge to aerial locomotion. Elevated ocean surface temperatures generated atmospheric instability that produced convective storm systems of considerable intensity, particularly over open water and along coastlines, where thermal gradients between land and sea drove strong directional winds. For a flying animal whose wing loading the ratio of body mass to wing area determined its minimum flight speed and its sensitivity to atmospheric turbulence, storm conditions represented a direct survival threat. A pterosaur caught in strong turbulence over open water, far from a coastline that offered a landing surface, faced the dual hazard of structural wing stress and exhaustion from continuous corrective flight effort. Thermal columns rising from sun-heated terrain provided the energetic basis for soaring flight during stable atmospheric conditions. A pterosaur capable of locating and exploiting thermal lift could gain altitude with minimal muscular effort, then glide across considerable horizontal distances before intercepting the next thermal column. This soaring strategy, used by modern large birds including albatrosses, vultures, and condors, is energetically efficient over long distances but requires sufficient wingspan and appropriate wing geometry to function effectively. The larger pterosaurs of the late Jurassic and subsequent Cretaceous period show skeletal proportions increasingly consistent with soaring specialization, suggesting that the exploitation of thermal and dynamic lift was a persistent, selective pressure shaping pterosaur wing evolution across the Mesozoic. The coastal cliffs and rocky outcroppings that bordered late Jurassic shallow seas provided both nesting substrate and aerodynamic infrastructure. Wind deflected upward along vertical cliff faces generates consistent lift in a narrow band above the cliff edge. Orographic lift that soaring animals exploit by flying parallel to the cliff face and remaining within the first. Ascending air column. Modern seabirds use this mechanism extensively along exposed coastlines, and the skeletal proportions of coastal pterosaur species are consistent with a similar behavioral strategy. From cliff edge launch points, pterosaurs could achieve immediate access to lift without the energetic cost of a ground-based takeoff run, a significant advantage for larger individuals whose mass made unassisted ground launch increasingly difficult. Aerial predation, where it occurred, would have been brief and precisely timed. A pterosaur descending from soaring altitude toward a prey animal at or near the water surface was converting potential energy gained through soaring into kinetic energy during the dive, arriving at the prey location with speed and momentum accumulated over a descent of potentially hundreds of meters. The precision required to intercept a moving fish at water surface during a high-speed approach placed significant demands on visual acuity and predictive motor control. Pterosaur orbit size, preserved in skeletal material, indicates large eyes relative to skull volume consistent with high-resolution vision and the capacity to track small, fast-moving targets at distance. The aerial ecosystem of the late Jurassic was not a refuge from the pressures operating at ground and sea level. It was a distinct physical domain with its own resource distribution, its own hazard profile, and its own selective pressures that shaped the animals inhabiting it over millions of years. Pterosaurs navigating this domain did so within constraints imposed simultaneously by atmospheric physics, prey availability, predation risk, reproductive requirement, and the fundamental energetic limits of powered and soaring flight. The sky above the Jurassic landscape was neither empty nor safe. It was occupied, contested, and demanding a third dimension of a world in which the pressure to perform precisely and continuously admitted no interval of biological complacency. During the late Jurassic, the configuration of the Earth's surface was fundamentally different from the present day. The supercontinent Pangaea, which had dominated global geography for tens of millions of years, had by this period fragmented into two primary landmasses, Laurasia in the north and Gondwana in the south, separated by the expanding Tethys Sea. These landmasses were themselves not uniform. They encompassed an extraordinary range of environmental conditions across their geographic extent, from equatorial floodplains receiving several meters of rainfall annually to interior continental regions where distance from oceanic moisture sources produced arid or semi-arid conditions across vast areas. Across this range of environments, the giants of the Jurassic had distributed themselves not uniformly, but in patterns that reflected the specific ecological requirements of individual lineages and the constraints imposed by geography, climate, and resource availability. The Morrison Formation of North America, which has yielded the largest concentration of late Jurassic dinosaur material from a single geographic region, preserves evidence of an ecosystem that extended across an area of more than 1 million square kilometers, roughly equivalent to the combined area of modern France, Germany, Spain, and Italy. This was not a single uniform habitat, but a mosaic of distinct environmental zones arranged across a gradient from the elevated volcanic highlands to the west, through extensive river floodplains in the central regions, to lower-lying coastal and lagoonal environments in the east, where a shallow inland sea periodically encroached from the north. Each of these zones supported distinct but partially overlapping communities of species, connected by the movement of animals across environmental boundaries. The floodplain environments that dominated much of the Morrison landscape were seasonally dynamic. Evidence from fossil soils, paleosols preserved within the Morrison stratigraphy, indicates alternating wet and dry seasons of considerable intensity. During wet seasons, river systems expanded across broad floodplains, depositing fine sediment across extensive areas and supporting dense vegetation growth along waterways. During dry seasons, these same rivers contracted to isolated pools and channels, concentrating animal populations around diminishing water sources and intensifying competition for both water and the food resources clustered near it. This seasonal cycle of abundance and scarcity was a primary organizing force in the Morrison ecosystem, driving the movement patterns, reproductive timing, and competitive interactions of the animals living within it. Isotopic analysis of paleosol carbonate from the Morrison Formation indicates mean annual precipitation estimates ranging from approximately 500 to 900 millimeters across different portions of the formation, with significant variability both spatially and temporally. These figures place the Morrison environment within a range comparable to modern semi-arid savanna or dry woodland, productive enough to support large herbivore populations during favorable periods, but subject to drought, stress severe enough to cause widespread plant, die-off, and water source concentration during dry extremes. The carrying capacity of this environment for large-bodied herbivores was therefore not constant, but fluctuated substantially across seasonal and multi-year timescales, with consequences for population dynamics that would have cascaded through the entire food web. On the southern landmass of Gondwana, equivalent ecosystems were developing in partial isolation from their Laurasian counterparts, The fragmentation of Pangaea had not yet produced the complete geographic separation that would characterize later periods, and faunal exchange between northern and southern landmasses occurred intermittently across land connections that formed and dissolved as sea levels fluctuated. The result was a global dinosaur fauna that showed broad taxonomic similarities. Sauropods, large theropods, and ornithischian herbivores were present on multiple continents, but with regional differentiation reflecting the degree of isolation and the specific environmental conditions of each landmass. In what is now Tanzania, the Tendaguru Formation preserves a late Jurassic fauna of remarkable diversity and scale, broadly contemporaneous with the Morrison Formation and sharing several closely related taxa. Giraffatitan branchi, formerly assigned to Brachiosaurus, reached estimated masses of 23,000 to 37,000 kilograms and represents one of the most completely known large sauropods from the African Jurassic. The presence of closely related sauropod genera on both the North American and African continents during the same geological interval confirms that faunal exchange was occurring across the Proto-Atlantic, which at this period remained sufficiently narrow and interrupted by island chains to permit occasional dispersal of large terrestrial animals between landmasses. The interior regions of both Laurasia and Gondwana, distant from the moderating influence of ocean moisture, experienced temperature extremes and precipitation deficits that fundamentally limited biological productivity. These continental interior environments were not entirely uninhabited. Fossil material from arid Jurassic deposits confirms the presence of small-bodied dinosaurs, early mammals, and a range of invertebrates capable of tolerating periodic desiccation, but they supported far lower biomass densities than the floodplain and coastal environments that concentrated large-bodied fauna. The distribution of giants across the Jurassic planet was therefore not global in the sense of uniform geographic coverage, but was instead concentrated in specific environmental corridors where water availability, vegetation, productivity, and climatic stability coincided sufficiently to sustain the energetic requirements of multi-ton animals across annual cycles. Volcanic highlands along the western margin of the Morrison landscape introduced a distinct set of environmental variables. Ash deposition from periodic eruptions altered soil chemistry across downwind areas, temporarily reducing vegetation productivity and potentially introducing toxic element concentrations including fluorine and sulfur compounds into water sources and plant tissue. Animals grazing on ash-contaminated vegetation or drinking from contaminated water sources would have experienced physiological stress proportional to the concentration and duration of exposure. Skeletal pathologies consistent with fluorosis, a condition caused by chronic fluorine ingestion that produces abnormal bone density and joint deterioration, have been identified in some large herbivore specimens from volcanically active Mesozoic regions, suggesting that proximity to volcanic activity carried measurable health costs for resident fauna. Migration across this varied landscape imposed its own physical demands. An animal of sauropod mass moving across terrain that included river crossings, elevation changes, and substrate transitions from hard rock to soft alluvial sediment was expending energy at rates that required proportionally abundant food resources along the route. The energetic economics of large body migration are considerably less favorable than those of smaller animals. The absolute cost per unit distance increases with body mass, even as locomotor efficiency partially compensates through longer stride length. For sauropods crossing hundreds of kilometers between seasonal resource patches, the quality and continuity of food resources along migration corridors was not incidental, but was a direct determinant of whether the energetic investment in movement yielded a net benefit. Trackway evidence confirms that sauropods moved across diverse terrain types and that their routes were not confined to optimal substrate. Tracks preserved in what were once hardened floodplain surfaces, river margins, and coastal mudflats indicate movement across a range of depositional environments, with substrate conditions varying from firm to saturated. The depth and morphology of individual tracks reflect both the mass of the animal and the mechanical properties of the substrate at the time of impression, providing indirect evidence of the conditions under which these animals were moving and the physical demands that variable terrain imposed on locomotion. The Jurassic planet, considered at a global scale, was not a world in which giants were exceptional. They were, across multiple continents and multiple environmental zones, the expected occupants of productive terrestrial ecosystems. Their presence shaped vegetation structure, soil composition, hydrology, and the behavior of every other species sharing their range. The question that the fossil record poses most insistently is not how these animals achieved their size, but how the ecosystems that produced and sustained them maintained the productivity and stability necessary to support biological structures of this magnitude across millions of years. The answer lies not in any single environmental variable, but in the intersection of atmospheric chemistry, tectonic configuration, seasonal hydrology, and the accumulated biological complexity of ecosystems that had been developing without interruption since the beginning of the Mesozoic. Survival in the late Jurassic was not a condition that, once achieved, could be maintained passively. It was an active, continuous process, one that required the uninterrupted functioning of biological systems operating under persistent external pressure. The fossil record, read carefully, does not present a picture of animals moving through an environment that was indifferent to their presence. It presents a picture of organisms whose bodies recorded, in bone and tissue, the cumulative evidence of a world that was actively and continuously testing the limits of their physiological capacity. Injury was among the most consistently documented conditions in the Jurassic fossil record. Healed fractures have been identified across a wide range of dinosaur taxa in skeletal elements including ribs, vertebrae, limb bones, and cranial material. The healing evident in these specimens confirms that the animals survived the initial trauma, but survival of a fracture in an environment of persistent predation, pressure, resource competition, and physical demand was not equivalent to full recovery of functional capacity. A large theropod with a healing rib fracture was simultaneously managing the metabolic cost of bone repair, the pain response associated with tissue damage, and the reduced locomotor or predatory performance that structural compromise imposed. In ecological terms, an injured predator was a less effective hunter, and a less effective hunter faced the compounding problem of reduced caloric intake at precisely the moment when metabolic demand was elevated by the repair process itself. For herbivores, injury carried a different but equally consequential set of implications. A sauropod with a compromised limb was less able to maintain pace with a moving herd, less able to accelerate away from a predator approach, and potentially less able to reach food resources that required any degree of locomotor effort. Skeletal evidence of healed injuries in sauropods includes pathological bone remodeling in vertebral elements consistent with compression fractures, injuries that may have resulted from falls on uneven terrain, from intraspecific contact during social interactions, or from predation attempts that made contact without achieving a fatal outcome. In each case, the injured animal continued to function. The healed bone confirms this, but did so under conditions of reduced capacity that elevated its vulnerability across every dimension of its ecological existence. Disease operated as a parallel and partially invisible pressure. Bone pathologies inconsistent with traumatic injury, including periosteal reactions, abnormal bone proliferation, and erosive lesions, have been identified in Jurassic dinosaur specimens and are consistent with bacterial infection, metabolic dysfunction, or neoplastic processes. The warm, humid conditions of the Jurassic forest environment were favorable to microbial activity, and an open wound sustained during predation or intraspecific conflict in this environment would have been exposed immediately to a dense community of bacteria and fungi. Without the immune sophistication of modern mammals or the behavioral capacity for wound care, injured dinosaurs depended entirely on their innate immune response to contain infection, a response that consumed metabolic resources and produced systemic physiological stress measurable in the growth record preserved in bone. Tissue. Starvation represented a seasonal and recurring threat whose signature is less directly visible in the fossil record than traumatic injury, but whose ecological significance was at least equivalent. The seasonal drought cycles documented in Morrison Formation paleosols would have produced periodic collapses in vegetation cover across large areas, reducing food availability for large herbivores at precisely the times when water stress was also most acute. Animals that entered a drought period with insufficient fat reserves, a condition more likely in juveniles, in socially subordinate individuals excluded from the best feeding areas, or in animals already compromised by injury or disease, faced a compounding sequence of physiological deterioration from which recovery was contingent on the return of favorable conditions within a time frame their reserves could sustain. Bone histology provides indirect evidence of nutritional stress in Jurassic dinosaurs. Growth rings in cortical bone, analogous in mechanism to the annual rings of trees, record periods of reduced or arrested growth that correspond to intervals of resource limitation. In some specimens, multiple growth rings are present, indicating that the animal experienced repeated seasonal growth interruptions across its lifetime. The width and density of bone deposited between growth rings reflects the quality of conditions during growth periods. Narrower, denser layers indicate slower growth consistent with marginal nutritional conditions, while wider layers of less dense bone reflect rapid growth under conditions of resource abundance. Reading this record across multiple specimens from the same formation allows reconstruction of the seasonal and interannual variability in resource availability that the living animals were navigating. Juvenile dinosaurs occupied a position of particular vulnerability within this system of pressures. Regardless of the eventual adult size of a species, all dinosaurs passed through a period of small body size during which the protective advantages conferred by mass were unavailable. A juvenile sauropod in its first years of life, before rapid growth had moved it beyond the prey size range of medium and large theropods, was exposed to predation pressure from a broader range of predators than the adult of the same species would ever face. Nesting sites, where eggs and hatchlings were concentrated in a fixed location, represented points of aggregated vulnerability detectable by predators through olfaction and accessible to a range of body sizes that the adult herbivores present could not effectively exclude. Evidence for predation on juvenile dinosaurs comes from multiple sources. Tooth marks consistent with small and medium theropod dentition have been identified on juvenile sauropod bones from Morrison Formation localities. The spatial association of small theropod material with sauropod nesting horizons in some deposits suggests that nesting areas attracted predators selectively, functioning as seasonally reliable food sources. For the nesting adults, the energetic and behavioral investment in nest defense represented a cost that had to be balanced against their own feeding and survival requirements, a trade-off that, in years of resource scarcity, may have been resolved in ways unfavorable to offspring survival. Intraspecific competition conflict between members of the same species imposed costs that are documented in the fossil record through pathologies concentrated in anatomically specific locations. In large theropods, bite marks consistent with conspecific dentition have been identified on cranial material, suggesting head-directed aggression between individuals of the same species. Hadrosaur and ceratopsian specimens from later periods show healed injuries on cranial display structures consistent with physical contact during intraspecific contests, and analogous evidence from Jurassic ornithischians indicates that this pattern of competitive physical interaction was not a late Mesozoic development, but a persistent feature of dinosaur social dynamics across the entire era. The resources over which intraspecific competition occurred included food, water, territory, and reproductive access, the same fundamental categories that drive competitive conflict in modern animal communities. During dry seasons, when water sources contracted to isolated pools and the vegetation surrounding them was heavily depleted, the concentration of animals at these points would have intensified competitive interactions to a degree not present during periods of resource abundance. An animal injured during intraspecific conflict at a water source was simultaneously dealing with the wound, the continued presence of competitors, reduced access to the water it had come to obtain, and the elevated predation risk that accompanied both injury and the concentration of multiple large animals in a small area. What the fossil record ultimately documents across thousands of individual specimens from Jurassic formations worldwide is not a world in which any species operated comfortably within its environment. It documents a world in which the bodies of animals were continuously marked by the pressures acting upon them, fractured and healed, infected and partially recovered, growth interrupted and resumed, worn by parasites and nutritional stress, and the physical demands of locomotion across difficult terrain. Survival in the late Jurassic was not a background condition. It was the primary biological output of every living system within the ecosystem, achieved imperfectly, temporarily, and at continuous cost, by animals whose bodies preserved in extraordinary detail the evidence of everything the world had required of them. The late Jurassic ecosystem was not defined exclusively by the animals whose size made them visible across geological time. Beneath the feeding arcs of sauropods, beneath the footfalls of large theropods, and beneath the canopy of a forest that was itself structured around the requirements of megafauna, an entire biological world operated at a scale that the fossil record has historically underrepresented, but which ecological modeling and increasingly fine-grained excavation techniques have revealed to be foundational to the function of the ecosystem as a whole. The small-bodied fauna of the Jurassic early mammals, lizard-like lepidosaurs, amphibians, insects, and a range of specialized scavengers were not peripheral to the ecosystem. They were structurally embedded within it, occupying roles that the large-bodied fauna could not fill and performing biological functions without which the broader system could not have operated. The energetic foundation of any terrestrial ecosystem is decomposition, the breakdown of organic material by microbial communities, fungi, and invertebrate detritivores that releases bound nutrients back into the soil system and makes them available for plant uptake. In the late Jurassic, this process operated at extraordinary scale. The biomass produced and shed by sauropod-dominated herbivore communities, including shedskin, feces, urine, and the carcasses of animals dying from injury, disease, or starvation, represented an enormous and continuous input of organic material into the decomposer system. Without the microbial, fungal, and invertebrate communities processing this material, nutrient cycling would have slowed, soil fertility would have declined, and the plant productivity that sustained the large-bodied herbivores would have been progressively undermined. Early mammals of the late Jurassic were more diverse in body plan and ecological role than their small body sizes might suggest. Multituberculates, a lineage that would persist well into the Cenozoic, showed dental morphology indicating omnivorous diets that included seeds, insects, and small vertebrates. Triconodonts possessed more carnivorous dental configurations, with interlocking cusps capable of processing invertebrate and small vertebrate prey. Docodonts, known from exceptionally preserved specimens including material from the Middle Jurassic Daohugu beds of China, show skeletal specializations indicating semi-aquatic, arboreal, and fossorial lifestyles within the same broader taxonomic group, a degree of ecological diversification that contradicts older models of Mesozoic mammals as uniformly small, nocturnal insectivores confined to a single ecological role. Castoricorda lutrasimilis, recovered from the Daohugu deposits and dating to approximately 164 million years ago, possessed a broad, flattened tail and limb proportions consistent with semi-aquatic locomotion, indicating that some Mesozoic mammals had already colonized aquatic foraging niches well before the end Cretaceous extinction. That is, traditionally framed as the starting point of mammalian ecological diversification, Volaticotherium antiquum from the same deposits preserves evidence of a gliding membrane, indicating arboreal locomotion and aerial dispersal capability. These specimens collectively demonstrate that the mammals of the Jurassic were not waiting passively for the extinction of the dinosaurs to diversify, they were actively exploring available ecological space within the constraints imposed by a world dominated by archosaurs. Lizard-like lepidosaurs, the lineage that today includes lizards, snakes, and the tuatara, were present throughout the Jurassic in forms that occupied insectivorous and small prey carnivore roles in forest floor and understory environments. Their ectothermic physiology, requiring less food per unit body mass than endothermic competitors of equivalent size, allowed them to persist through periods of resource scarcity that would have imposed greater stress on metabolically faster mammals. In the humid, thermally stable conditions of the Jurassic forest floor, where ambient temperatures remained within ranges compatible with ectotherm activity for much of the year, lepidosaurs competed effectively with mammals for invertebrate prey and filled ecological roles in the small-body predator guild that mammals had not yet fully occupied. Amphibians, frogs, salamanders, and the now extinct albinerpetontids were abundant in the moist environments of the Jurassic, particularly in association with rivers, lakes, and wetland margins where their reproductive requirement for standing water was met. Frog diversity expanded significantly during the Jurassic, with multiple modern frog families having origins traceable to this period. Their ecological role as mid-level consumers feeding on invertebrates and being fed upon by small reptiles, mammals, and larger amphibians placed them at a critical position in the food web, transferring energy from the invertebrate-dominated decomposer system upward into the vertebrate predator community. The invertebrate fauna of the Jurassic forest floor was itself a complete ecological system operating largely beneath the perceptual threshold of the large-bodied animals above it. Beetles, whose diversity has made them the most species-rich animal order in modern ecosystems, were already diversifying rapidly during the Jurassic, occupying decomposer, herbivore, predator, and parasite roles within the litter and soil community. Termite-like social insects were present. Though the highly derived termite lineages of the modern era had not yet appeared, wood decomposition was instead carried out by a range of beetle larvae and other invertebrates whose collective processing of dead plant material was a primary mechanism of carbon and nutrient cycling in the forest. Ants, which would later become dominant social insects in terrestrial ecosystems, had not yet appeared in the Jurassic. Their origin is placed closer to the mid-Cretaceous. However, other social or semi-social insects occupied analogous ecological roles, and the structural complexity of the invertebrate community was sufficient to support multiple trophic levels of invertebrate predation. Centipedes, scorpions, and predatory beetles occupied positions as apex predators within the litter community, preying on smaller invertebrates and occasionally on the eggs or hatchlings of small vertebrates whose nests were accessible at ground level. Scavenging as an ecological strategy was likely more prevalent and more systematically organized in the Jurassic than the fossil record directly documents. Large carcasses, sauropods, dying from drought, disease, or predation represented food resources of enormous caloric value that would have attracted a succession of consumers at different body sizes and with different anatomical tools for accessing different tissue types. Large theropods capable of breaching the thick skin and subcutaneous tissue of a sauropod carcass would have been the primary openers, exposing internal organs and muscle tissue to a subsequent community of smaller consumers. Small theropods and ornithischians could access exposed soft tissue. Mammals and lepidosaurs could penetrate into cavities and extract material inaccessible to larger animals. Invertebrates, particularly fly larvae and beetle communities, would complete the process, reducing remaining organic material to a state where microbial decomposers could finalize nutrient release. This successional scavenging sequence ensured that the caloric value of a large carcass was extracted with high efficiency across multiple consumer levels, minimizing waste and maximizing the transfer of nutrients from dead biomass back into the living system. The small-bodied fauna were not merely incidental participants in this process, they were the agents of its completion, responsible for the final stages of a nutrient cycling sequence whose earlier stages depended on the large-bodied predators and herbivores that have historically received the majority of paleontological attention. The hidden fauna of the Jurassic, small, cryptic, operating at the margins of detection for both their contemporaries and for the paleontologists who would later attempt to reconstruct their world, were not diminished versions of the giants above them. They were differently scaled expressions of the same evolutionary pressures, occupying ecological space with the same precision and the same vulnerability, and performing biological functions whose importance to the stability of the overall ecosystem was inversely proportional to the size of the animals performing them. The giants of the Jurassic were visible. The hidden creatures were foundational. Between them, they sustained a biological system of extraordinary complexity across tens of millions of years, one in which no component, however small, was ecologically expendable. The surface of the late Jurassic Earth was geologically active in ways that had direct and measurable consequences for the biological systems operating above it. The fragmentation of Pangaea, the tectonic process that was progressively separating the planet's landmasses during this period, was driven by the movement of crustal plates across a mantle whose convective currents generated zones of persistent volcanic activity along plate boundaries, at mid-ocean ridges, and above mantle hotspots where concentrated thermal energy was transferred upward through the crust. This volcanic activity was not episodic in the sense of being rare or exceptional. It was a structural feature of the Jurassic Earth, operating continuously at varying intensities across multiple geographic regions simultaneously, and its outputs, gases, ash, lava, and hydrothermal fluids entered the atmosphere, the hydrosphere, and the soil system in quantities sufficient to influence conditions across continental scales. The most geologically significant volcanic activity of the late Jurassic was concentrated along the developing rift zones where Laurasia and Gondwana were separating, and along the subduction boundaries where oceanic crust was being forced beneath continental margins. Along the western margin of what is now North America, subduction-related volcanism was building the precursors of the mountain systems that would eventually become the Sierra Nevada and associated ranges. This activity produced both explosive eruptions that injected material into the upper atmosphere and effusive lava flows that resurfaced portions of the landscape at intervals determined by the underlying tectonic dynamics. The geological record of the Morrison Formation preserves ash layers, bentonite beds at multiple stratigraphic horizons, confirming that explosive volcanic events were depositing material across the Morrison landscape throughout the period of its formation. The atmospheric consequences of sustained volcanic activity operated across multiple timescales. Individual explosive eruptions injected sulfur dioxide into the stratosphere, where it combined with water vapor to form sulfate aerosols that reflected incoming solar radiation and produced measurable surface cooling lasting months to years. These short-term cooling events would have disrupted plant growth cycles, reduced the productivity of photosynthetic communities, and compressed the food resources available to large herbivore populations during the period of reduced insolation. For animals already navigating seasonal resource scarcity, a volcanically induced growing season disruption represented an additional and unpredictable source of nutritional stress superimposed on the regular annual cycle. On longer timescales, volcanic outgassing of carbon dioxide contributed to the elevated atmospheric CO2 concentrations characteristic of the Jurassic period. The mid-ocean ridge systems actively expanding during Pangaea's fragmentation were particularly significant sources of CO2 outgassing, as fresh basaltic crust exposed along ridge crests reacted with seawater and released stored carbon. This sustained input of CO2 into the atmosphere maintained the greenhouse conditions that kept global temperatures elevated above modern baselines and supported the high plant productivity that the sauropod-dominated herbivore communities depended upon. Volcanic activity was therefore simultaneously a source of short-term ecological disruption and a long-term driver of the environmental conditions that made the Jurassic ecosystem possible. Lava flows presented the most immediately destructive form of volcanic impact on local ecosystems. Where effusive eruptions produced fluid basaltic lava, flows could advance across terrain at rates of several meters per hour under favorable slope and viscosity conditions, incinerating vegetation and rendering the substrate uninhabitable for decades to centuries until weathering and biological colonization processes gradually restored soil development. Animals in the path of an advancing lava flow had limited options. Those capable of rapid locomotion could relocate, while slower moving or less mobile fauna were eliminated within the affected area. The ecological consequence was not merely the loss of individual animals, but the destruction of habitat structure, the physical framework of vegetation, burrow systems, and substrate complexity within which the biological community was organized. The recovery of ecosystems following volcanic disturbance followed a successional sequence whose timescale depended on the nature and extent of the disturbance. Fresh basaltic substrate, once cooled, was initially colonized by pioneer microbial communities and then by spore-dispersing plants, ferns, and horsetails whose reproductive biology allowed rapid establishment on bare mineral surfaces. This early colonization created organic matter inputs that accelerated soil development, eventually permitting the establishment of the gymnosperm communities that dominated Jurassic forest ecosystems. The entire successional sequence from bare lava to mature forest, likely required centuries to millennia depending on climate conditions and the proximity of seed and spore sources. During this interval, the recovering area supported reduced biomass and limited structural complexity conditions that constrained the re-establishment of large-bodied fauna and created extended periods of reduced carrying capacity across affected landscapes. Volcanic gases presented a more pervasive and chemically diverse threat than lava flows alone. Sulfur dioxide, hydrogen sulfide, carbon dioxide, and hydrogen fluoride were among the primary gaseous outputs of Jurassic volcanic systems, and each carried distinct toxicological implications for biological systems at different concentration ranges. Carbon dioxide, denser than air, accumulates in low-lying terrain near volcanic vents and can reach concentrations lethal to animals within enclosed valleys or depressions without producing prior warning through odor or visible change. Hydrogen sulfide, with its characteristic odor detectable at very low concentrations, was potentially more behaviorally avoidable, but at concentrations above threshold, produced rapid incapacitation that eliminated the behavioral response it would otherwise trigger. Fluorine compounds introduced into soil and water systems through volcanic deposition produced long-term biological effects, extending well beyond the immediate vicinity of eruptive events. Fluorine is incorporated into plant tissue at concentrations reflecting the geochemical composition of the soil from which the plant draws nutrients. And animals ingesting fluorine-contaminated vegetation over extended periods accumulate the element in skeletal tissue, a condition producing the bone and joint pathologies characteristic of fluorosis. The spatial extent of fluorine contamination following a volcanic ash deposition event could encompass thousands of square kilometers, affecting animals that had no direct contact with the volcanic source but were consuming vegetation grown on contaminated substrate across a broad downwind area. Hydrothermal systems associated with volcanic activity modified local hydrology in ways that influenced animal behavior and habitat use. Hot springs and geothermal seeps maintained elevated water temperatures year-round, creating microenvironments of distinctive chemistry and thermal character that some organisms exploited while others avoided. Mineral precipitation around hydrothermal vents produced distinctive substrate types, silica-rich sinters, carbonate mounds, that altered local drainage patterns and created conditions of unusual geochemical composition in associated soil and water. Animals drinking from hydrothermal water sources were ingesting elevated concentrations of dissolved minerals whose physiological effects depended on concentration and duration of exposure. The interaction between volcanic systems and the hydrological cycle extended to river chemistry across entire drainage basins. Rivers sourced in volcanically active highland regions carried elevated loads of dissolved minerals and suspended volcanic material that altered the chemistry of water across their entire downstream extent. Aquatic organisms in these systems, fish, amphibians, invertebrates, were exposed to geochemical conditions that fluctuated with volcanic activity upstream, and the productivity of riparian and wetland ecosystems downstream was partially determined by the mineral inputs delivered by volcanically influenced river systems. The geological violence of the Jurassic was not always visible at the surface. Much of it operated through chemical and atmospheric pathways that left no immediate dramatic signature but exerted continuous influence on the biological systems above. The volcanoes of the late Jurassic were not external events that occasionally intersected with the ecosystem. They were embedded within the same planetary system that produced the climate, the soil chemistry, the atmospheric composition, and the tectonic configuration that define the conditions of Jurassic life. The ecosystem and the geology were not separate phenomena operating in parallel. They were components of a single integrated Earth system, each continuously influencing the other across timescales, ranging from the instantaneous violence of an individual eruption to the 100-million-year arc of continental fragmentation that restructured the planet's surface. Throughout the Mesozoic, as the fragmentation of Pangaea progressed through the Jurassic period, one of its most ecologically consequential outcomes was not the separation of land masses itself, but the creation of the shallow marine environments that formed between them, where continental crust rifted apart and subsided below sea level. Seawater flooded the resulting basins, producing epicontinental seas of variable depth, chemistry, and connectivity. These were not the deep, uniform oceanic environments of fully separated ocean basins. They were geologically young, physically dynamic, and biologically distinctive environments in which marine life was colonizing newly available habitat while simultaneously being shaped by the specific chemical and physical conditions that the young seafloor and adjacent landmasses imposed. The Proto-Atlantic, forming as North America and Europe separated along a developing mid-ocean ridge, was during the late Jurassic still a relatively narrow and shallow seaway in its northern portions. Its southern extension, connecting to the Tethys Sea, provided a marine corridor that linked the developing Atlantic basin to the broader global ocean. Along its margins, carbonate sediments accumulated in warm, shallow water, the geological record of reef-building organisms, shell-producing invertebrates, and the skeletal remains of marine vertebrates that inhabited these productive coastal environments. The lateral extent of these carbonate platforms, preserved today in limestone sequences across Europe and eastern North America, reflects the biological productivity of environments that were, for their time, among the most ecologically diverse on Earth. Shallow epicontinental seas possess physical characteristics that distinguish them fundamentally from both deep ocean environments and terrestrial ecosystems. Their limited depth, often less than 200 meters across extensive areas, means that solar radiation penetrates to or near the seafloor across much of their extent, supporting benthic photosynthetic communities and the invertebrate fauna dependent on them. Temperature variability is greater in shallow water than in the deep ocean, as seasonal changes in solar input and atmospheric temperature are transmitted more directly to the water column. Salinity can vary significantly near river mouths and in semi-enclosed basins where evaporation exceeds freshwater input. These physical gradients of light, temperature, and salinity created a mosaic of microenvironmental conditions across the shallow Jurassic seas, each supporting a distinctive assemblage of organisms adapted to its specific parameters. Reef-building organisms of the Jurassic differed substantially from the coral-dominated reef systems of the modern ocean. Scleractinian corals were present and contributed to carbonate framework construction, but the primary reef builders of many Jurassic carbonate platforms were sponges, specifically calcareous and siliceous sponges that formed the structural matrix of reef complexes across much of the Tethyan region and the developing European shelf seas. These sponge microbial reefs supported extraordinary invertebrate diversity, including brachiopods, bivalves, echinoderms, and a range of mobile predatory invertebrates that exploited the structural complexity of the reef framework. The ecological architecture of these communities, the spatial organization of filter feeders, grazers, predators, and detritivores across the reef structure was functionally analogous to modern reef ecosystems, despite the substantial taxonomic differences in the organisms involved. The isolation of marine basins produced by continental fragmentation had evolutionary consequences that operated on geological timescales but were already detectable within the Jurassic period. As seaways narrowed or deepened, as land connections formed or dissolved with changing sea levels, populations of marine organisms were periodically separated into distinct basins with limited genetic exchange. Under these conditions, populations diverged through the accumulation of genetic differences in isolation, a process that continued over millions of years, produced the regional differentiation in marine fauna, visible in the fossil record of different Jurassic basins. The Tethys Sea, which extended as a broadly east-west tropical seaway between Laurasia to the north and Gondwana to the south, supported a marine fauna of high diversity maintained by the stable, warm temperatures and high productivity of its tropical waters. Species distributions within the Tethyan realm show geographic patterning consistent with the influence of current systems, depth barriers, and salinity gradients on dispersal, confirming that the sea was not a uniform environment permitting free exchange of organisms across its extent, but was instead structured by physical gradients that created partial barriers to dispersal and promoted regional biological differentiation. Coastal predators of the Jurassic shallow seas exploited the high productivity of nearshore environments through a range of feeding strategies. Marine crocodilians, teleosauroids, and metriorhynchids were present in Jurassic seas in forms substantially more adapted to marine life than their modern freshwater relatives. Metriorhynchids had lost the heavy osteodermal armor characteristic of terrestrial crocodilians, reducing body density and improving hydrodynamic efficiency. Their limbs showed modification toward paddle-like forms, and their tails were adapted for the lateral undulation that drives aquatic locomotion in fish-like swimmers. These animals occupied a predatory role in shallow coastal environments, pursuing fish and cephalopods in the water column while remaining capable of terrestrial movement, though likely with reduced efficiency compared to their freshwater relatives. Hybodon sharks, a lineage that had been present since the Paleozoic, maintained significant diversity in Jurassic marine environments, occupying a range of ecological roles from hard prey crushing specialists to active pursuit predators of fish and cephalopods. Their teeth, the most commonly preserved part of the shark skeleton, show morphological diversity consistent with dietary partitioning across species, and their widespread occurrence across multiple Jurassic marine formations indicates that they were ecologically ubiquitous in both shallow epicontinental seas and open marine environments. The presence of multiple shark lineages alongside marine reptiles in the same depositional environments confirms that the shallow Jurassic seas supported overlapping predator communities in which competition for prey resources was a persistent ecological reality. Sea level change, driven by the combined effects of tectonic activity and the thermal expansion of seawater under elevated Jurassic temperatures, periodically altered the geography of shallow marine environments at timescales ranging from thousands to millions of years. Rising sea levels flooded low-lying continental margins, expanding the area of shallow marine habitat and connecting previously isolated basins. Falling sea levels exposed shallow seafloor as dry land, eliminating marine habitat across extensive areas and isolating populations in residual basins whose reduced connectivity promoted rapid evolutionary divergence. These sea level cycles were not smooth and continuous, but episodic, driven by pulses of tectonic activity along mid-ocean ridges and by the periodic melting or growth of polar ice in a world where polar ice was substantially reduced relative to modern conditions, but not entirely absent during all intervals of the Jurassic. The biological consequences of sea level change extended beyond the marine realm. When sea levels rose sufficiently to flood low-lying terrestrial corridors, Land connections between landmasses were severed, isolating terrestrial populations and initiating the same divergence processes operating in the marine environment. When sea levels fell, new land connections formed, permitting faunal exchange between previously isolated terrestrial regions. The alternation between connected and isolated states, driven by the same geological processes that were reshaping the ocean floor, produced a planetary-scale system of biological fragmentation and reconnection that generated evolutionary novelty across both marine and terrestrial ecosystems simultaneously. The strange seas between continents were strange precisely because they were new, geologically young environments whose physical characteristics were still being determined by the tectonic processes creating them, and whose biological communities were still being assembled from the pool of organisms capable of colonizing and persisting in conditions that had no prior equivalent. The organisms inhabiting these seas were not simply marine organisms doing what marine organisms had always done. They were participants in an ongoing ecological experiment whose parameters were being continuously modified by forces operating at the scale of the planet itself, forces that neither the organisms nor the ecosystems they constituted had any capacity to anticipate or control, but to which they responded, generation by generation, through the only mechanism available to biological systems under environmental pressure: the slow, imprecise, and relentless process of evolutionary change. The late Jurassic was not a static endpoint. It was a transitional interval, a period during which the ecological structures, species compositions, and environmental conditions that had characterized the Middle Jurassic were progressively modified by forces operating across biological, geological, and atmospheric dimensions simultaneously. These changes did not announce themselves through sudden catastrophic events. They accumulated gradually, expressed through shifts in species distribution, changes in ecosystem productivity, and the slow replacement of established taxa by new lineages whose anatomical and physiological innovations positioned them more effectively within the conditions that were emerging. The world of the late Jurassic was, in measurable ways, already becoming something other than what it had been, and the direction of that change pointed toward a Cretaceous period whose biological character would differ from the Jurassic in ways that only the accumulated evidence of millions of years could fully document. Climate instability during the late Jurassic is documented in the sedimentary record through several converging lines of evidence. Paleosol sequences from the Morrison Formation show alternating horizons of calcrete indicating dry, well-drained conditions and horizons of organic-rich mudstone consistent with waterlogged, poorly drained soils. This alternation reflects not a gradual directional shift in climate, but an oscillating pattern of wet and dry intervals whose frequency and amplitude appear to have increased through the late Jurassic relative to earlier periods. The driving mechanisms behind this instability included the progressive reconfiguration of ocean circulation patterns as the developing Atlantic basin altered the distribution of oceanic heat transport, and changes in the reflectivity and moisture retention of the land surface as vegetation cover responded to shifting precipitation regimes. Ocean circulation changes consequent to continental fragmentation had atmospheric implications that extended far inland. As the Proto-Atlantic widened and deepened, the exchange of water between the developing Atlantic basin and the Tethys Sea established new current systems whose heat transport properties differed from those of the restricted, shallow seaways they replaced. Regions that had previously been buffered against temperature extremes by proximity to warm, shallow seas found themselves increasingly exposed to continental interior conditions as the seas withdrew. Conversely, Newly formed coastlines experienced the moderating influence of oceanic proximity for the first time, producing local climate regimes with no prior analogue in the region's geological history. These shifts in regional climate were not rapid enough to constitute discrete events in the ecological experience of any individual organism, but they were consequential over the timescales of species persistence and ecosystem assembly. Vegetation communities responded to these shifting conditions through compositional change that the fossil pollen and spore record documents with increasing detail. The gymnosperm-dominated forests of the mid-Jurassic were not replaced abruptly, but their species composition shifted, certain genera declining in relative abundance, while others expanded into the ecological space made available by changing moisture and temperature regimes. More significantly, the earliest representatives of the angiosperm lineage, the flowering plants that would come to dominate terrestrial vegetation in the Cretaceous and Cenozoic, appear in the fossil record in proximity to the angiosperm lineage. Jurassic-Cretaceous boundary. Though their initial ecological impact was limited, the appearance of angiosperms represents one of the most consequential botanical transitions in Earth history. But its beginning was quiet, a minor element in a plant community still dominated by conifers, cycads, and ferns, detectable in the fossil record only through careful analysis of dispersed pollen assemblages. The faunal changes visible across the Jurassic-Cretaceous transition were not uniform in character or rate across different taxonomic groups. Some lineages that had been ecologically dominant through the Jurassic declined substantially in diversity before the boundary, suggesting that the environmental changes of the late Jurassic itself, rather than events at the boundary, were the primary drivers of their reduction. Others show no significant change across the boundary, their species ranges and ecological roles continuing without major disruption into the early Cretaceous. Still, others, particularly among the smaller-bodied vertebrates and the insects, show patterns of diversification accelerating through the late Jurassic and into the Cretaceous, suggesting that the same environmental changes that were challenging established large-bodied taxa were simultaneously opening ecological opportunities for lineages with greater physiological flexibility or shorter generation. Times. Among the most ecologically significant transitions occurring across this interval was the diversification of Maniraptoran theropods, the dinosaur clade that includes the ancestors of modern birds. By the late Jurassic, multiple Maniraptoran lineages had evolved, and the iconic Archaeopteryx lithographica, preserved in extraordinary detail in the Solnhofen limestone at approximately 150 million years ago, documents a stage of avian evolution in which flight capability, feathered integument, and a mosaic of ancestral and derived skeletal features coexisted in a single animal. The ecological implications of powered flight access to aerial food resources, rapid dispersal across geographic barriers, escape from ground-level predation, positioned the early avian lineage for the diversification that would eventually produce the most species-rich class of terrestrial vertebrates in the modern world. In the late Jurassic, this diversification was only beginning, but its foundations were already in place. Competition between established and emerging lineages intensified as environmental conditions became less stable and resource distribution became less predictable. In ecosystems where conditions are stable over long periods, competitive relationships between species tend toward equilibrium states in which established taxa maintain advantage through familiarity with local conditions, accumulated behavioral knowledge, and the ecological infrastructure territory, social networks, learned resource locations that experienced individuals possess. When conditions shift, these advantages are partially eroded. Novel conditions favor physiological generalism, rapid reproductive response, and behavioral flexibility over the specialized adaptations that confer competitive advantage in stable environments. The late Jurassic, as a period of increasing environmental variability, was therefore a period in which the competitive landscape was being partially reset, in which the advantages that had sustained dominant taxa were being selectively devalued and replaced by a different set of biological doing great. Currencies. The response of large-bodied sauropods to these changing conditions is not straightforwardly interpretable from the fossil record. Sauropod diversity in the late Jurassic was high, with multiple lineages coexisting across multiple continents. Across the Jurassic-Cretaceous boundary, some lineages disappear from the record, while others persist and diversify into the Cretaceous. The Neosauropod clade, which includes the titanosaurs that would become dominant large herbivores of the Cretaceous, shows continuity across the boundary, suggesting that the physiological and ecological characteristics of this lineage were compatible with the conditions of both the late Jurassic and the early Cretaceous, even as related lineages declined. Whether this persistence reflected specific adaptive advantages or simply the probability outcomes of survival under conditions that were challenging but not uniformly lethal across all large herbivore taxa, remains an active area paleontological investigation. The beginning of a changing world was not experienced as change by the animals living within it. The timescales of ecological and evolutionary transition, thousands to millions of years, are incommensurable with the timescales of individual biological experience. Each generation of late Jurassic organisms was born into a world that differed imperceptibly from the world of the preceding generation, navigated the pressures of that world with the biological tools inherited from ancestors who had faced similar pressures, and produced offspring whose inherited characteristics would be tested against conditions that were, generation by generation, incrementally different from those of their parents. The change was real, and its cumulative consequences were profound. But it was change at the pace of geology, expressed not in the experience of individuals, but in the composition of populations, in the distribution of species across landscapes, and in the slow replacement of one ecological configuration by another. The late Jurassic was ending not through catastrophe, but through the accumulation of small shifts whose direction was not predetermined and whose outcome, from within the system, was entirely invisible. What the fossil record reveals, from the outside and across deep time, is a world in the process of becoming something else, still recognizably Jurassic in its dominant taxa and ecological structure, but already carrying within its biological and geological fabric the seeds of a transformation whose full expression would require millions of years more to complete. The boundary between the Jurassic and Cretaceous periods, placed by current stratigraphic convention at approximately 145 million years ago, was not marked by the kind of abrupt, globally synchronous event that defines other major boundaries in the geological timescale. There was no mass extinction of the magnitude associated with the end Permian or end Cretaceous boundaries, no global iridium anomaly indicating a bolide impact, no single stratigraphic horizon that preserves the simultaneous disappearance of dominant taxa across multiple continents. What the geological record shows instead is a transition prolonged, geographically variable, and taxonomically selective in which the ecological configurations of the late Jurassic were progressively dismantled and reassembled into the different but equally complex structures of the early Cretaceous. This transition was not instantaneous. It unfolded across several million years of accumulated biological and environmental change, and its expression differed substantially between regions, between taxonomic groups, and between environmental settings. The sedimentary record spanning the Jurassic-Cretaceous boundary is discontinuous across many of the formations that preserve late Jurassic fauna. In the Morrison Formation, the uppermost sedimentary layers show evidence of significant erosion before the deposition of overlying Cretaceous strata, a stratigraphic gap that represents an unknown but potentially substantial interval of time during which deposition was either absent or subsequently removed. This gap complicates the direct documentation of faunal change across the boundary in North America, making it difficult to distinguish between taxa that disappeared at or before the boundary and those whose absence from Cretaceous deposits reflects the incompleteness of the record rather than genuine extinction. The distinction matters because these two patterns imply different causal mechanisms and different rates of ecological change. Environmental stress in the latest Jurassic is documented through multiple independent proxies. Carbon isotope records from carbonate sequences spanning the boundary interval show excursions consistent with disruptions to the global carbon cycle intervals during which the relative abundance of carbon isotopes in marine carbonates shifted in ways that reflect changes in oceanic productivity, organic carbon burial rates, or the input of isotopically distinct carbon from volcanic or terrestrial sources. These carbon cycle perturbations were not as severe as those associated with the major mass extinction boundaries, but they were real, and they indicate that the global biogeochemical systems regulating ocean chemistry and atmospheric composition were experiencing instability during this interval. Sea level change across the Jurassic-Cretaceous boundary was significant and geographically variable. In some regions, regression, the withdrawal of shallow seas from continental margins, exposed extensive areas of previously marine habitat as dry land, eliminating the productive shallow water ecosystems that had supported high marine diversity through the Jurassic. In other regions, transgression brought seawater onto previously terrestrial areas, fragmenting continuous land surfaces and isolating populations of terrestrial organisms. These sea level changes were driven by the continued tectonic reorganization of the planet's surface, the same processes that had been reshaping continental configuration throughout the Jurassic and their biological consequences operated across the same slow timescales as the tectonic forces producing them. The large sauropod lineages that had defined the ecological character of the late Jurassic did not disappear uniformly at the boundary. Diplodocoids, the lineage including Diplodocus, Apatosaurus, and their relatives, declined substantially across the boundary and are absent or extremely rare from early Cretaceous deposits in most regions. Whether this decline reflected competitive displacement by other herbivore lineages, habitat change that reduced the availability of vegetation types compatible with diplodocoid feeding morphology, or simply the stochastic outcomes of population reduction under deteriorating conditions, cannot be fully resolved from the existing record. What is clear is that the ecological role occupied by large diplodocoid sauropods in the late Jurassic was not immediately or completely filled by equivalent animals in the early Cretaceous. There was a period of restructuring during Tempass, which the large herbivore guild of terrestrial ecosystems was being reorganized around different taxa with different body plans and different feeding ecologies. Titanosaur sauropods, by contrast, persisted across the boundary and diversified substantially in the Cretaceous, eventually achieving a near-global distribution and producing some of the largest individual animals in the terrestrial fossil record. The characteristics that distinguished titanosaurs from their diplodocoid relatives, including more robust skeletal construction, different vertebral architecture, and potentially different feeding reach and dietary preferences, appear to have been compatible with the environmental conditions of the Cretaceous in ways that the diplodocoid body plan was not. The nature of this compatibility is not fully understood, but it represents one of the clearest examples of differential survival across a major temporal boundary within a single broadly defined ecological guild. Theropod diversity across the Jurassic-Cretaceous boundary shows a different pattern from that of sauropods. Large allosauriod theropods, the group including Allosaurus and its relatives, declined toward and across the boundary, with the clade becoming substantially reduced in diversity in the early Cretaceous relative to its late Jurassic peak. Their ecological role as large apex predators was progressively assumed by members of the Coelurosaurian lineage, the broader group that includes tyrannosauroids, dromaeosaurids, and ultimately birds whose diversification through the latest Jurassic and early Cretaceous tracked the decline of the Allosaurids with a degree of temporal correspondence that suggests competitive dynamics were involved. Though the evidence does not permit confident attribution of causation, the Stegosaurid lineage, which had been a characteristic component of late Jurassic herbivore communities across multiple continents, declined sharply across the boundary. Stegosaurus and its close relatives disappear from the fossil record in the earliest Cretaceous, and while isolated Stegosaurid material from slightly later Cretaceous deposits has been reported from some regions, the lineage was clearly in significant decline. The ecological factors driving Stegosaurid decline are not well constrained. Their feeding morphology, adapted for low browsing on soft vegetation, may have been disadvantaged by changes in vegetation composition, or competitive pressure from ornithopod dinosaurs whose diversity and abundance was increasing through this interval may have reduced the resources available to Stegosaurids below the threshold necessary to sustain viable populations. The final generations of the iconic Jurassic giants moved through landscapes that were, in their gross physical appearance, still recognizably Jurassic forested, warm, and populated by animals of extraordinary size. But the composition of those forests was changing. The species present within them were shifting, and the ecological relationships that had organized the community for tens of millions of years were being slowly renegotiated under conditions that no longer perfectly replicated those under which they had developed. Individual animals were not aware of this transition. They were navigating the immediate pressures of survival, finding food, avoiding injury, reproducing with no access to the broader temporal perspective that reveals their position within a historical arc of biological change. What the fossil record preserves of these final Jurassic generations is fragmentary, as it is for all geological intervals, but it is sufficient to confirm that the transition was not sudden for the organisms living through it. Species that would eventually disappear from the record continue to be present, continue to interact with their ecological community, continue to leave the traces, bones, tracks, tooth marks that would be recovered and interpreted by investigators working 145 million years later. Their extinction was not an event that those final generations experienced. It was a pattern that emerged from the accumulated outcomes of individual lives, each of which ended through the ordinary mechanisms of Jurassic mortality, in a world that was becoming, without announcement or awareness, the early Cretaceous. The giants of the Jurassic did not vanish into silence. They entered the rock. Over millions of years, the physical remains of animals whose living mass had shaken the ground beneath them were reduced through the sequential action of biological, chemical, and geological processes to mineralized structures preserved within sedimentary sequences that accumulated above them. The transformation from living organism to fossil is not a simple or guaranteed process. The vast majority of organisms that have ever lived left no recoverable trace. What survives in the fossil record represents a small and systematically biased sample of past biological diversity weighted toward organisms with hard skeletal structures, toward environments with high rates of sediment deposition, and toward geological settings where subsequent erosion has re-exposed buried material without destroying it. Understanding what the fossil record shows requires understanding, with equal precision, what it does not show and why. The process of fossilization begins at the moment of death, when the biological systems maintaining an organism's structural integrity cease to function and decomposition begins. In most cases, soft tissue is consumed by scavengers, broken down by microbial communities, or dispersed through the environment before any preservation can occur. Skeletal material, bone, teeth, shell, is more resistant to decomposition but is subject to physical disarticulation by scavengers and water currents, to chemical dissolution in acidic soils, and to mechanical destruction by trampling or geological stress. Exceptional preservation of articulated skeletons, soft tissue impressions, or gut contents requires a specific convergence of conditions: rapid burial in fine-grained sediment, low oxygen concentration, inhibiting microbial decomposition, and geochemical conditions promoting the replacement of organic material with stable minerals before structural integrity is lost. The Solnhofen limestone of Bavaria represents one of the most extraordinary examples of exceptional preservation in the Jurassic fossil record. Deposited in a lagoonal environment of unusually high salinity conditions that reduce the diversity and activity of scavenging organisms, the Solnhofen sediments accumulated as fine carbonate mud that buried organisms with sufficient rapidity and chemical neutrality to preserve anatomical details invisible in coarser depositional environments. Feather impressions, wing membrane outlines, and the soft tissue contours of organisms that would otherwise be known only from their skeletal elements are preserved in these deposits, with a resolution that has made them disproportionately influential in the reconstruction of late Jurassic biology. Archaeopteryx, known from multiple Solnhofen specimens, would be interpretable as a theropod dinosaur indistinguishable from its non-avian relatives. On skeletal evidence alone, it is the feather impressions, preserved only because of the exceptional depositional conditions of the lagoon, that reveal its position at the base of the avian lineage. The Morrison Formation presents a contrasting taphonomic environment, one of high fossil abundance, but generally lower preservation quality than the Solnhofen. The concentrated accumulations of dinosaur material in Morrison quarries reflect depositional processes that brought large numbers of skeletal elements together, drought-related mortality at water sources, river transport of carcass material, and the activity of scavengers concentrating bones in specific microenvironments rather than in situ preservation of complete articulated individuals. Reading the behavioral and ecological information encoded in these deposits requires distinguishing between the original biological signal and the taphonomic processes that modified it between death and discovery. A concentration of Allosaurus material at a single locality may reflect predator trapping, seasonal aggregation, or simply the hydraulic sorting of bones by river currents, and determining which explanation is correct requires analysis of bone orientation, weathering stage, and sedimentological context that goes beyond the presence of the bones themselves. The recovery of Jurassic paleontology as a scientific discipline has been neither linear nor uniform. Major discoveries have restructured understanding of Jurassic biology in ways that earlier investigators could not have anticipated, and the pattern of discovery reflects the geographic distribution of research effort as much as the actual distribution of fossil material. The Morrison Formation received intensive investigation from the late 19th century onward, beginning with the competitive excavations of the Bone Wars period, in which the rival paleontologists Othniel Charles Marsh and Edward Drinker Cope drove the description of dozens of new Jurassic dinosaur species through a combination of genuine scientific productivity and mutual antagonism that produced errors of identification and attribution that required decades of subsequent revision to correct. The Chinese Jurassic fossil record, by contrast, was substantially less known to Western science until the late 20th century, when systematic excavation of deposits including the Yanliao biota of Inner Mongolia and the Shishugu Formation of Xinjiang began producing material that fundamentally altered understanding of Jurassic dinosaur diversity and biogeography. The Yanliao biota, dating to approximately 160 million years ago, has yielded feathered dinosaurs, early mammals with diverse locomotor specializations, and flying vertebrates whose anatomical characteristics provide critical information about the sequence of evolutionary innovations leading to powered flight. These discoveries confirmed that the evolutionary transitions previously inferred from later Cretaceous material were already underway in the Middle Jurassic, compressing the apparent timeframe of avian origin and revealing a degree of early Mesozoic biological diversity that the Morrison-centered research tradition had not predicted. Biomechanical analysis of Jurassic dinosaur material has progressively replaced qualitative morphological description as the primary tool for reconstructing the functional biology of extinct animals. Finite element analysis, a computational technique that models the distribution of stress within a structure under applied loads, has been applied to digitally reconstructed dinosaur skulls to evaluate competing hypotheses about feeding mechanics, bite force, and the behavioral contexts in which specific skeletal features were used. Muscle reconstruction, based on the identification of attachment scars on bone surfaces and comparison with the musculature of living archosaurs, provides the force inputs for these biomechanical models and allows quantitative predictions about the locomotor and predatory performance of animals known only from their skeletal remains. Isotopic analysis has opened additional windows into the biology of Jurassic animals that skeletal morphology alone cannot provide. Oxygen isotope ratios preserved in tooth enamel and bone apatite reflect the temperature of body fluids during mineralization, providing evidence about thermoregulatory physiology that contributes to ongoing debates about metabolic rates in dinosaurs and other Mesozoic vertebrates. Carbon isotope ratios in the same materials reflect dietary inputs. The isotopic signatures of different plant types are preserved in the tissues of animals consuming them, allowing dietary reconstruction at a level of detail unavailable from tooth morphology alone. Strontium isotope ratios, whose geographic variation reflects the geochemical composition of local bedrock, can track the movement of individual animals across landscapes during their lifetimes, providing direct evidence of migration or dispersal in animals that left no behavioral record in any other form. The landscapes where Jurassic giants once moved are now exposed as eroded badlands, cliff faces, and river valleys where the sedimentary sequences deposited during their lifetimes have been re-exposed by millions of years of subsequent geological activity. Walking through the Morrison Badlands of Utah or Wyoming, or across the limestone plateaus of the Tendaguru region in Tanzania, is to move through a physical environment whose surface expression reflects processes operating across deep time. The uplift of ancient sedimentary basins, the erosion of overlying strata, the differential weathering of rock types deposited under conditions that no longer exist anywhere on the planet's surface. The fossils emerging from these landscapes are not simply objects of scientific interest. They are physical connections to a biological world whose complexity and scale the geological record continues, incrementally, to reveal. What the Jurassic ultimately demonstrates, across the full breadth of evidence now available from its fossil record, is that the biological world is capable of organizing itself into configurations of extraordinary complexity, scale, and internal coherence, and that these configurations, however stable they appear across the timescales of human observation, are temporary expressions of conditions that are themselves continuously changing. The giants that walked the late Jurassic landscape, the predators that hunted them, the small mammals that survived beneath their feet, the marine reptiles that patrolled the shallow seas between fragmenting continents, all of these were participants in a biological system that operated with precision and interdependence for tens of millions of years before the slow accumulation of geological and environmental change began to dismantle it. The stone remembers what the living world forgot. And in recovering that memory, fragment by fragment, formation by formation, the science of paleontology continues to reconstruct a world that was, in its time, as complete and as real as the one that has succeeded it.